Can somebody confirm you can hear me?
Yeah, we can hear you.
Thank you. All right, before we begin, are there any questions? I'm going to get some water. My voice seems like it's a little hoarse. Sorry about that. I guess I haven't talked today, so I didn't notice my voice was off. Um, are there any questions before we begin? Sharing my screen now. All right, we had talked about culture media last time. Media is to support microbial growth. A medium has to provide an energy source, a carbon source, sources of nitrogen, sulfur, phosphorus, trace elements, and organic growth factors if an organism needs these factors. Most microbes will not need an organic growth factor added to the media. Um, Streptococcus pyogenes would, but most others, like E. coli, would not. You do, however, need for E. coli to have a carbon source, an energy source, a source of nitrogen, sulfur, and phosphorus, and then trace elements, which can come usually just from the water. But here's a question for you. If you have a photoautotroph that can fix nitrogen, what do we need to supply from the media? Do you need to supply an energy source? That's a yes or no question, people.
I say yes.
Only if you're not supplying light. If it's a photoautotroph and you're in the sunlight, it doesn't need an energy source. How about a carbon source? Does a photoautotroph need a carbon source? The answer is no, because it can pull CO2 from the air. Now, I said it fixes nitrogen, so it doesn't need a source of nitrogen. So all you need to supply in this media would be a sulfur source, a phosphorus source, and trace elements. Any question about any of that? I think we've talked a little bit about agar before. Agar is a complex polysaccharide derived from seaweed. It's used as a solidifying agent in our culture media to make it a solid. We usually put agar in a tube. We have two types, a slant and a deep. Let me see if I can show that. Nope, not getting paint here. Let's try that again. There it goes. So if we have a tube like this and pour the agar in, and it's a flat, we call that a deep. If on the other hand you have a tube like this, and the auger is put at a slant, like that, we call that a slant. Any question about any of that? Agar is generally not metabolized by microbes. It was actually Dr. Koch, when he was growing microbes on plates, a solid plate, he initially was using jello. And the trouble with jello is some microbes could digest the jello, making the plate a liquid. And that would make a mess out of his solid culture. And it was actually a wife of one of his assistants who suggested, why don't you use agar? Agar is a substance you use in baking or thickening, like when you're making a sauce. And he tried it and it worked very well. And it worked so well that we're still using it as the primary agent we put in a liquid broth to make it a solid. So usually when we're talking about a solid media, we're using agar, but you could use jello or gelatin, and you can also use starch. Those are the three agents we have for making a media a solid. Any question about any of that? Like I said, agar is not metabolized by microbes, and so the plate or the tube will remain a solid. We have different types of culture media. One broad type is a chemically defined media. Here we know the exact chemical composition of the media because all the chemicals we've added have been things that we added, if that makes sense. Here's a very simple chemical defined media. Let me see if I can blow this up. All this media has in it is glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and then water. And so these are the only molecules in the media. This worked very well for a non-fastidious organism that can grow in a general purpose media. This general purpose media, which one is this? This one would be a glucose salts media. However, if you have a fastidious organism, you got to add more ingredients, something like this. So we got glucose, starch, sodium acetate, sodium citrate, oxaloacetic acetate, or oxaloacetate, a bunch of different elements, amino acids, organic growth factors, etc. This is an unusually chemically defined media because it has so many different ingredients like uracil, biotin, but this is still a chemically defined media because all of the ingredients we're adding. So we know what all of the molecules are in this media. Most complex media contain extracts from digestive yeast, meat, or plants, and these digestive yeast, meat, or plants provide vitamins and organic growth factors, but the exact chemical composition of a complex media is not known. For example, here's a simple complex media that we call nutrient agar, or Na. It has peptone, which is a partially digested protein from soybean, 5 grams, beef extract, so 3 grams of dried beef, sodium chloride, agar, and then water. We don't know the exact chemical makeup of this media because depending on what the cow had been eating, you're going to be adding different molecules. And that could also be different for the where you grow the soybeans. You're going to pick up different molecules in the soybeans. Any question about that? So a complex media, the exact chemical composition is not known. Peptone is a partial protein digest, and it's the primary energy source, carbon source, nitrogen source, and sulfur source of this media. Although you can get energy, carbon, nitrogen, and sulfur also from the beef extract. But there's more peptone in this media. Complex media are frequently used when you're growing heterotrophs, bacteria and fungi. They require an organic carbon source, and that will be supplied by the beef extract and the peptone. Any question about any of that? So all of the general purpose media that we've discussed, tryptic soy agar, nutrient agar, and nutrient agar with yeast extract, is a complex media. Any question about any of that? All right, we have different types of culture media. Obviously, we have general purpose media, we have chemically defined media, and then complex media. We also have anaerobic media. This is important media for growing obligate anaerobes. Obligate anaerobes must avoid the exposure to oxygen because oxygen will kill the cells. It has a reducing compound in it, meaning a chemical like sodium thioglycolate that combines with and removes oxygen from the media. And so a thioglycolate media will have oxygen depletion, at least in the bottom of the media. You heat the media to drive off any absorbed oxygen, and then the sodium thioglycolate will remove the oxygen from the media. Any question about any of that? We also have selective media. This has chemicals in it that halt or inhibit the growth of a particular group of microorganisms, but not affecting the growth of another group of microorganisms. An example of a selective media is 7% sodium chloride media. Most bacteria cannot grow in 7% sodium chloride. They do not grow above 2%, most bacteria. But there are some bacteria that can grow in 7% sodium chloride. So 7% sodium chloride is a selective media, allowing you to select for microbes that tolerate salt. Any question about any of that? We also have a differential media. This media has chemicals in it that allow you to show differences between different groups of organisms. These differences are not related to how well the organism grows on the medium. You may be able to tell the differences by different color of the microbes growing on the media or by a different result that the microbe causes to the media. But it makes it easy to distinguish between colonies of different microbes. An example of a differential media is blood agar plates. In blood agar plates, you can differentiate between organisms that have no hemolysis. It's called gamma hemolysis, where the blood agar is not lysed all the way up until you have the microbes on the plate. And then you can distinguish that between alpha hemolysis, that is partial hemolysis, where the blood agar, let me see if I can blow this up a little, where the blood agar is partially hemolyzed before the microbes. If we're looking at the red here, we notice a darkening of the red right before the off-white of the alpha streak of the microbes. And inside the alpha, it's all darkened. This darkening, which is usually either brownish or blackish in color, and sometimes it's also greenish in color, is from the partial lysing of the red blood cells by this microbe. Any question about any of that? Let me shrink that down again now. And that can be distinguished from this one here, which is beta hemolysis or complete hemolysis, where when we look at the red blood cells here, we notice a region of clear around the streak of the beta, which is a white microorganism, and inside the beta, clearing of the blood, where there is no red because all of the blood cells have been lysed. You can actually hold that plate up and see through the beta hemolysis. I like to either look at a light bulb or a clock. Like I said, you can see right through the beta hemolysis. You'll notice that beta hemolysis has a darkened ring around the beta hemolysis. So that darkened ring is actually alpha hemolysis, and that's something that's normally seen with beta hemolysis, meaning the red blood cells are red up until the darkening, which is alpha hemolysis, and then you go in further, you have beta hemolysis, and there is no more blood. Any question about blood agar? We also have a selective and differential media. This media is both selective and differential. An example of a selective and differential media is EMB, or eosine methylene blue. It is selective against gram-positive bacteria, so if you get growth on the plate of EMB, then it's most likely to be gram-negative bacteria growing, because gram-positives tend not to grow. But it's also differential on the basis of lactose fermentation. If the bacteria can grow on the plate, it will be either a metallic greenish color, kind of like this color here, or a black from the fermentation of the sugar lactose. It changes the color to either black or metallic green. And if you have a non-lactose fermenter and it's a gram negative, it'll grow on the plate, but it will not be metallic green or black in color. It'll be the bacteria's normal color, which in this case would be white. Any question about that? Now remember, on a selective and differential media, if you do not have growth on the plate, you cannot assay for the differential ability of the bacteria. Meaning if you had a gram positive and it did not grow on EMB, you cannot tell whether the bacteria is a lactose fermenter or not, because you have no growth to assay. Any question about any of that? There's another type of media which we call an enrichment media. It is a media that encourages the growth of a certain group of microbes. So it is selective in nature. So all enrichment media is also a selective media. We just use the enrichment media a little differently because I don't know, we're not using it exactly as a selective media. We're using it to select for microorganisms of growth. And what happens is it will increase their numbers of a desired microbe to detectable levels. It's particularly useful if the targeted microorganism is only a small proportion of the total population. And it's often used in enrichment media when you're trying to grow certain microbes from the soil, other environments, or from a fecal sample. Suppose you get a fecal sample and you're testing for a microbe that may cause disease. But the trouble is, the disease causing organism is at low numbers, at least in the fecal sample. So what you can do is grow it in an enrichment media and then select for the microbe of interest. An example of that would be, let's say we're looking for a phenol digesting microbe that we obtain from the soil. So soil does have some phenol digesting microbes in it, but the trouble is there's thousands or maybe even hundreds of thousands of other bacteria in the soil. And so if we were to streak out uh, colonies, you would have to streak out over 1,000 colonies just to get one colony of interest, meaning a colony that could actually digest phenol. So what you can do is put it in an enrichment media where we select for the bacteria of interest. So if we're interested in a phenol digesting microbe, we can put it in a media where phenol is the only carbon source and the only energy source. This will select for phenol digesting microbes. Let me go back a little. Let me end this show and start here. Paint again. Can't get that for some reason. It keeps going to that first time. Second time is going the right place. So let's say we had soil. Or soil sample. But the phenol, oops, the phenol digesters are like 1 in 1,000,000. I don't know if I can do that. I can't do that here. Let's do the old-fashioned way. Can't make that probably a superscript here. Ah, let's just go 2, 3. Well, we can put it in a enrichment media that will select for phenol. And let's say it enriches 100-fold. Now, this will mean that the phenol will then be 1 over 10 to the 4th, which means we'd need to streak out to get one colony of interest, 10,000 colonies. That would be way too much for anyone to do. But what we could do is, get my arrow back, take a sample from this tube and put it in another enrichment tube. Oh, not drawing that very good, but that's all right. And let's say it enriches 100-fold again. Now we have 1 in 100 colonies. We could streak out 100 colonies and get of interest 1 colony of interest. So this would be doable after you've grown it up in two enrichment tubes, meaning you grow it up in the first and then transfer it to the second. We've now increased the phenol digesters to 1 in 100 cells. So if we were to streak out 100 different colonies, one of them would be a colony of interest, meaning a phenol digester. And that could be done. There'd be a lot of colonies to streak out, but it could be done. Any question about how we use an enrichment media? All right, this table is giving you the different type of culture medias that I just talked about, so it's a summary. Let's talk a little bit about obtaining a pure culture. A pure culture is a culture containing only one species. A colony is a population of cells arising from a single cell or two cells or a group of attached cells in a clump, meaning more than one cell. Therefore, when we talk about colonies on a plate, we frequently use the term colony forming unit because some colonies are formed by one cell, but about 30% of the colonies on a plate are formed by more than one cell. And 30% of the colonies not being pure is not good enough in microbiology. How you obtain the single colony is usually by streaking for colony isolation, called the streak plate method, which I usually call streaking for colony isolation. Let me blow this slide up. What you do is you separate the plate into sectors or quadrants, and then streak it out. That's not too bad. You want about 20 to 30 streaks, which is actually shown here, in each sector. It's not shown here, but you want about 20 to 30 streaks. And then you sterilize your loop. Make my mouse the loop. So put it in the flame or the incinerator. And then start out sector 2. You need to go into sector 1 about 3 times, and this has done it excessively. It's gone 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Like I said, you only want 3 or 4 times to go into sector 1, and then streak it out 20 to 30 more times, and then sterilize the loop and bring it back to sector 3. It's not shown here, but we can assume that they're streaking this way, and they didn't pick up anything to begin with. And then go here. What's important about both of this one and that one is you do not, when you're starting sector 3, you do not want to go back where your microbes are the heaviest in sector 2. And this will be where they're the heaviest. You want to go where they're lighter. In fact, this is probably a little too far. We should only be going in about this far. You do want to come in about 3 times into sector 2. And this really looks like it's only gone in one time. I mean, they probably did something like this and then that. And you do want it to go in more than once. You want it to go in about 3 times. So this is a little excessive. 1, 2, 3, 4, 5, 6. That's not too bad. Oh, wait a minute. There's 2 right here too. But the point is you don't want to go where the bacteria are most dense. When you're streaking out sector 1, be the most dense where you start and the least dense at the end. You only want to get in the bacteria where there's lower bacteria in sector 1 to make sector 2. Yeah, this is going in a little too far, but it's all right. It's better with sector 3. because that looks like it ends there. And then someplace along the plate, you'll get isolated cells or clumps of cells put down, and they'll grow into isolated colonies. Here, we're getting isolated colonies in sector 3. But if you look closely, you're starting to get isolated colonies in sector 2. They're a little too close together. You really want them separated further. And if you had a really dilute solution of bacteria to begin with, you could even get your isolated colonies in sector 1. Obviously, that didn't happen here. Anyways, that's the purpose of using the streaking for colony isolation, so that you eventually get someplace on the plate isolated colonies. Now, the thing is, you have to have the cells of interest in large enough numbers to the total microbial population. If that's not the case, you need to grow the culture in an enrichment media first. And when you get an isolated colony, that doesn't mean that that will be a pure colony. As I stated, 30% of these colonies are being formed by more than one cell. So they do not look like they are multiple species in the colony, but you don't know that until you've streaked it out again. So what you can do is take out an isolated colony here and then streak it out on a second plate for colony isolation. Let me see if I've got another picture of that. Nope, I don't. So streak it out on a second plate for colony isolation. Even then, you can't say with 100% clarity that your culture is now pure, because on the first plate, let's say these are two different plates here. Here, 30% of the colonies will be formed by more than one cell. You streak it out again on a second plate. Well, it's 30% and 30%. That reduces the number of colonies that are formed by more than one cell down to about 5% or less. So even if you streak it out for colony isolation 2 times, you cannot say with 100% purity that all of the colonies on the second plate are from a pure culture. The only sure way that you can tell if you have a pure culture is streak out a colony, and then if all the colonies look alike on the plate, then it's a pretty good indication you have a pure culture. Obviously, this plate shows that this was not a pure culture, because we have 2 colony types here. That doesn't mean 100%, but it's very high that if all the colonies look alike, then you have a pure culture. It's probably over 99%. Anyways, my point is, if you streak out your stock culture and pass the cells through 2 colony isolation events, meaning you first streak it out one time and you get one isolated colony, and then you streak it out a second time and you take the cells from an isolated colony that's gone through 2 colony isolation events, then those cells are most likely to be a pure culture. It's not 100%, but it's over 95%. And that's good enough in microbiology. Any question about any of that? All right, when we're talking about the growth of bacterial cultures, you have to remember that we're talking about the number of cells, not the cell size. Bacteria can grow by three different means, depending on the species. The vast majority of bacterial species grow by binary fission, where the cell literally splits into two, where one cell will split into two to form two cells. There are a few bacteria that can grow by budding. Cells form a small outgrowth on the parent cell that we call a bud. The bud enlarges, and then it'll separate from the parent cell when the bud becomes about the same size as the parent cell. I'll show you a little picture of that in just a minute. And then there's a few species of bacteria that can reproduce by reproductive spores. These reproductive spores usually are at the tips of a filament. Let me go ahead and draw that out now. Come on. Ah, there we got it. So we have a filament bacteria. That's just a long, it might not be a bacilli, but I'm drawing it that way. The bacilli tends to stand up, and then when it makes a reproductive spore, the spores tend to be small things on one edge of the bacteria, the filament. Well, let's make one. Well, I got four there. That's good enough. Here we started with one cell, and then after reproduction, we now have five cells, the parent cell, and then the four reproductive spores. These reproductive spores are smaller than the parent cell, and they're at one end, and that way a stiff wind could separate the spore, or maybe some animal will go by, pick up a spore, and then deliver it elsewhere. The reproductive spore can then grow into a parent cell. The point is, this is reproduction because we had one cell, and then after reproduction, we have five cells. So that's obviously reproduction. That differs from an endospore, where one parent cell makes one endospore, and then the parent cell dies. So 1-to-1 is not reproduction. An endospore is not a reproductive strategy. It is a survival strategy. A reproductive spore is a reproductive strategy. Any question about any of that? All right, let's move back. Going through binary fission, we have one cell which will first divide its DNA, and then the DNA will move towards opposite ends of the cell. That cell will then grow a plasma membrane and a cell wall between the cells down the middle, 50-50, and dividing the two strands of DNA. And the cell membrane and the cell wall will literally grow to split the cell into two. Here you can see, let me blow that up. This is an actual electron microscopic image of a cell undergoing binary fission, where the cell wall is just beginning to be formed in the cell. And that looks like a little bit of the cell membrane there. Any question about any of that? And then those two divide, become two independent cells, and then can even split and then go elsewhere. They don't have to go elsewhere, but they then split. Any questions about binary fission? Most bacteria divide by binary fission. A few bacteria divide by budding, where the parent cell here, excuse me, the parent cell here puts out a bud, and then that bud can enlarge and enlarge. Let's look at a case of budding. No commercial this time. So here we're looking at the cell putting out a bud right there. Let me bring this down. Right there, you can see the DNA start to divide. And there the DNA is moving into the bud. The bud is enlarging. And about right there, the bud is now separated from the parent cell and it becomes its own cell, no longer a bud. And there, both this parent cell is putting out a new bud there, and this new cell is putting out a bud there. Any questions about any of that? When we're talking about bacterial growth, you should come to realize that bacteria cells have a generation time. The generation time is the time required for a cell to divide into two, or it's the same as the population number in doubling in time. So when one cell splits and goes into two, that's the generation time of one cell. And for the population, it would be for the population number to double in size. The generation time varies considerably between different species. It can be as fast as 20 minutes. And we say, like the E. coli can have a generation time of 20 minutes. We mean that the generation time will be 20 minutes per generation. Most bacteria have a little slower generation time, where the generation time will be 1 to 3 hours per generation. The per generation is often left off because it's implied. Mycobacterium leprae, the bacterium that causes leprosy, I should say one of the bacterium causing leprosy, has a generation time of about 10 days. This is the slowest generation time that I know of. And it's because the mycobacterium make a lot of mycolic acid and put it in their cell wall. And it takes time for the bacterium to make mycolic acid. Remember that the generation time is the time per generation. Students frequently get the generation time wrong because I guess they're so used to talking about miles per hour, like when you're driving. This isn't time, this isn't generations per unit of time. It's time per generation. That's the generation time. Any question about any of that? All right. When we're talking about bacteria growing, we frequently, at least microbiologists, frequently like to plot it on a semi-log plot, where the generations are on the X scale, and this is an arithmetic scale, 0, 5, 10, 15, 20. But the Y number is a logarithmic number, and this is the number of cells. If we use a log scale, we will then get a linear relationship, or I should say a semi-log scale. We'll get a linear relationship with the growth. When you use a log scale, you also separate out the points down here at the start. And then the next point here, you can extend the graph, and the next point will go about here. If you were to plot that same plot on an arithmetic scale, where the x-axis is still arithmetic, and the number of cells is arithmetic, you have a whole bunch of points in the start. So one cell will divide into two, 2 into 4, 4 into 8, 8 into 16, 16 into 32, meaning all of these points are all clumped together right here. Furthermore, the next point, you can't extend the graph because it's not going to be here. It's going to be on the ceiling. So there's no way to graph the next point on this piece of paper. And then another reason why not to use an arithmetic scale is because this equation for this curve is much more complex than the equation for this curve. This curve is simply y, that's the number of cells, equals mx, m being a constant, x being the number of cells, plus b, b being the point of intersection on the graph, where I think it's where the y is going to equal 0. This one would be a much more complex equation, something like y equals mx plus b plus nx squared plus c, something like that. Any questions about why microbiologists like to use a semi-log plot? All right. When we're talking about bacteria growth, it's important to realize that there are different phases of growth. And you can usually find this happening for all organisms, not just for microbes. Here we're looking at the bacterial growth curve when we take a population of cells from the solid media and put it into a liquid media. But if you take a population of deer and put the deer on an island, we will get this similar growth curve. And they've done this, I don't remember where, but some island on the east coast of America. They put a bunch of deer on an island and found the deer population did this growth curve. So when you inoculate cells from a solid media into a liquid media, initially you'll have a lag phase where the cells are not growing. What the cells are doing in the lag phase is getting prepared for growth in the new media, meaning they're making the enzymes and other molecules that they need to grow in the new media, which in this case is a liquid media. Once they have the enzymes and the molecules they need for growing in the media, they then enter the log or exponential growth phase. Initially, the growth will be very rapid. The reason is initially there's lots of nutrients around, very low, if non-existent waste products around, and lots of space around, very few cells around. So the cells will grow very rapidly initially in the exponential growth phase. But as you get the population to get older, the growth will start to slow. Why the growth slows is now there's less space around the cells and more cells around the cells because the population density is higher. There's also less nutrients around and more waste products around. So the growth will begin to slow. Eventually, the growth will slow to the point where the population numbers will remain the same, and that we call the stationary phase. The population number stays the same because the number of cells growing equals the number of cells dying. The lucky cells are growing because they have space around them, at least some space around them, some nutrients around them, and some waste products, but not so many waste products that the cells cannot grow. The unlucky cells, on the other hand, don't have much space around them, don't have much nutrients around them, and they have lots of waste products around them. And these cells will begin to die. So in the stationary phase, the number of cells growing equals the number of cells dying. Why the population is remaining stationary, meaning the same number. But as the population ages, you'll get a buildup of more waste products. You get a decrease in the nutrients and also a decrease in the number of free space. So the population will then enter the death or decline phase, where the number of cells growing is less than the number of cells dying. So the total population of cells in the population starts declining. Now, early in the death phase, you may have some cells, which are very lucky, which are still capable of growing. You just have more cells that are dying. But late in the decline or death phase, decline or death phase, you have no cells growing. You only have cells which are remaining, I don't know, inactive, meaning they're not dying, but they're not reproducing, or you have cells which are dying. So late in the death phase, there's no cells which are reproducing. How long this death phase will last will depend on the species. A bacteria species like E. coli will not remain in the death phase for too long. After 1 or 2 weeks, all the cells of E. coli will die. You won't have anything left, alive at least. But if you have something that's a little hardier, like Staphylococcus aureus, it can remain alive longer. I'm not sure how much longer, but let's just say longer. And then if you have a species that can make an endospore, this death phase can continue for millennia. I think the oldest endospore is something like 40,000 years old. They dug it up from a ice sample they drilled out of a core of ice in Antarctica, and it was 40,000 years old. Of course, it was frozen too. That helped it live. But they then thawed it out and noticed the cell becoming metabolically active and then going on to reproduce. The point is that if the cell produces endospores, it can be in the death phase for a very, very long time. Any question about any of that? All right. When we're talking about microbes, it's important to discuss the different ways we have for measuring the growth of microbial populations. There are direct ways we can get the cell numbers and indirect methods we can get the cell numbers. The direct methods include a plate count, also called the viable plate count, the filtration method, the most probable number, a direct microscopic count, and then the indirect methods involve turbidity, looking at the turbidity of the microbes in the solution. You can also get an indirect method by the metabolic activity of the cells, or you can do a dry weight. Grow up your cells, centrifuge them down, pour off the liquid, allow the cells to dry, usually put them under a vacuum and maybe a little bit of heat to get them to dry, and then weigh the cells. Any question about any of those? I've used all of these methods for counting cells except for the most probable number method. Planck counts are the most common method used in microbiology to get an estimate of the number of bacteria in a solution. The plate count measures the number of viable cells that are capable of forming a colony on a plate. So the plate counts are often called a viable plate count because you're only counting the number of viable cells. The dead cells don't get counted. A plate count takes 24 hours or more for the colonies to form, and the results are often reported not as the number of cells per mil, but the number of colony forming units per mil. The reason being is that we get a number of colonies on a plate, but we do not know if those colonies are formed by one cell, two cells, or a clump of cells. So we report it as the number of colony forming units per mil. To get a plate count, you first make a serial dilution of a original sample. Let's say you're sampling bacteria from the soil, so you put, 1 gram of soil in 1 ml of water, and then you make serial dilution of that soil sample, meaning you take 1 ml out of the soil sample and put it into 9 mls, mix the cells up, and then take 1 ml out of that now 10 mls and put it into 9 more mls. Sort of like here. Original inoculum, which would be 1 gram of soil and 1 ml of water. You take 1 ml, put it into 9, so you've now got 1 and 10. Take 1 ml of this, put it into 9. Now you have 1 to 100. Take 1 ml of this, put it into 9 mls. Now you got 1 to 1,000 solution, 1 to 10,000, and then 1 to 100,000. You then plate out the cells on a plate using either the pour plate method, where you actually pour a mill on the plate and then spread out that mill, then allow the water or whatever to be absorbed, and then incubate the plate to get the number of colonies. Or the spread plate method, where you're taking 0.1 of a mil of your original solution, spreading it out on the plate, and then incubating it to get your individual colonies. Now, when you do this, you will get something looking like this. Here they didn't do the original inoculum, but they got 1 to 10 dilution, and there's too many colonies to count. And then there's the 1 to 100 dilution. It's also too many to count. The 1 to 1,000 dilution, we're starting to be able to count it, but we can't count them in large regions of the plate. So it's still too many colonies to count. And then we have the 1 to 10,000 dilution. And here we can count the colonies. And then the 1 to 100,000, where we can also count the colonies. Um, you want to count the colonies on a plate with 25 to 250 colonies on the plate. And then we report that dilution as 25 to 250 colony forming units per mil. So for example, right here, we have on this plate 4 colonies. So we can say that the number of cells in this dilution are 4 colony forming units per mil. Any question about any of that? This one, it looks like there's 30 colonies. I'm going to just guess 30 colonies for this dilution. And we report that as 30 colony forming units per this dilution. Now for this one, which was 4, you take that number and then multiply by the inverse of the dilution. So the dilution is 1 in 100,000. So we'd take 4 times 100,000. This one would be 30 times 10,000. So this one we'd say 30,000 colony forming units in the original inoculum. This one we'd say 40,000 colonies in the original inoculum. We want to take the count of the colonies between 25 and 250 colonies per plate. So we want to take our count of the original inoculum from this plate here. And I did point out that they got 2 different numbers. That would be like 40,000. This would be 30,000. We want to use the 30,000 per mil because the standard deviation of this plate for the mean is much less than that plate. Let me see if I can demonstrate that. On the plate where we have 4 colony forming units per mil, if we have plus or minus 1 colony, why didn't that work? Oh, I got it. No, I don't. There, I got it. It's plus or minus 25% on your mean. Everyone see that? So if you have plus or minus 1 colony from 4, you got plus or minus 25% for your mean. If on the other hand, you had 30 colonies forming units per mil, and you go plus or minus 1, it's not plus or minus 25%. What the heck is that? Something like 3.3%, is that right? Let me do a check there because nobody's saying anything and I don't remember. 1 divided by 30. Oop, I gotta go equals beforehand. Yeah, it's a plus 0.33%. So I was right, 3.3% for your mean. 3.3% is a lot smaller standard deviation than 25%. Everyone see that? So you want to take for which plate to base your mean on, you want to get it from this plate because the mean will be a lot more accurate than this plate. Any question about any of that? All right, the filtration method is similar to the plate count. The difference is we have a very dilute sample of bacteria to begin with. So dilute that if you were to streak it out or do a dilution, even the original inoculum will not give you enough colonies to get a good number. And what you do is you put that solution through a filter, where the filter will grab on and hold on to the bacteria, which cannot go past the filter. And then you take this filter and you put it on a plate of media and then grow the colonies on the plate. So here it looks like we have about 40 colonies. Let me see if I can blow that up. Looks like about 40 colonies on the plate. And let's say you did this filter and it's a whole gallon, and we have 40 colonies per gallon. Let's say you're sampling water, like drinking water. So the number of colony forming units per gallon is only 40. Any question about the filtration method? You use the filtration method only when your bacterial sample is very dilute. And it's similar to the plate count method, meaning you're actually counting up cells that form a colony. The difference is you're not diluting to get the colonies, you're actually increasing the colonies to grow on the plate by passing the liquid through a filter. Any question about any of that? If not, I'm gonna. Oh, we got a few more minutes. I thought it was 20. It's actually 10. All right, let's go on. All right. You can also get a direct measure of the number of microbes by counting the cells under a microscope. This is usually done by putting a volume of liquid in a gridded slide, and then the cells are counted under the grid. This method is not good for cells that are motile because, let's blow this up, if the cells are motile and you're counting the cells in this square here, this cell could move out of the square. And then let's say you counted this row here, and then that cell moves into that row. So it doesn't work well with motile cells. It's a little more difficult to do if the cell has a capsule, but it is possible to do it with a capsule. You're going to be counting both viable live cells and dead cells, because when you're looking at the cell under a microscope, you will not be able to tell whether the cell is viable or whether it is dead. You need a fairly high concentration of cells to use the microscopic count method, but if you have a really dilute sample of cells, you could always centrifuge them. Like I stated on this one, what you do is you take a measured volume of liquid and put it under the slide. The gridded slide is usually set up so that the volume of liquid under this one grid here is 1 microliter. And so you would take this number here and multiply by 1,000,000 to get the number of cells in the original solution. So here, what is that, 15 to 20? 14, I was right, 14. So you take this 14 times 1,000,000, so 14 million cells per mil in the original solution. And it's just that the volume under this grid there will be 1 microliter. And the volume under the gridded slide is definitely designed to be a certain volume. And usually they're 1 microliter for this grid there, and then you count the cells under this grid here. Anything, questions about that? Now, one problem with the direct microscopic count is you can't tell if this cell is alive or if it's dead. Another problem is for the poor schmuck doing the count, it's a little hard on their eyes. One advantage of this method is you get the count pretty quickly. I'm not going to talk about the most probable number method, but we're going to talk about the indirect methods now. There's 3 indirect methods where you indirectly get a number, an estimate of the cells: the turbidity, the metabolic activity, and the dry weight. Turbidity is using a liquid culture, and you compare the light coming through the media with the light coming through the media containing the cells. The cells will block some of the light from getting through, and then you measure how much light is getting through with a turbidity meter. Using a spectrophotometer. You then look on a chart to see the absorbance of the culture to get the number of cells. So initially, someone has to go through and have a certain dilution of cells, like 1 to 100, 1 to 1,000, 1 to, well, not that, excuse me. You'd have to have a certain number of cells per mil. You would have something like probably 1,000 cells per mil, 10,000 cells per mil, 100,000 cells per mil, 1,000,000 cells per mil, if I got that right. And then you measure the number of cells per mil and look at the absorbance, meaning how much light is being blocked. And then you make a chart. The very first time, it's going to be very time consuming to make the chart. But once you have the chart, you can then use that chart indefinitely. And you just compare the light getting through the media with the light getting through the cells, and then look up on the chart what the cell concentration is. And that's how you get an indirect method for the number of cells by using turbidity. Any question about any of that? A metabolic activity assumes that a bacteria's metabolic product will accumulate in direct proportion to the number of bacteria cells. So if you're measuring the amount of CO2, the more cells there are, the more CO2 there will be. The first time you do the metabolic activity, you have to have a number of cells, and you make different dilutions of the number of cells. So there would be something like 100 cells per mil, 1,000 cells per mil, 10,000 cells per mil, 100,000 cells per mil. And then you measure the amount of CO2 per the number of cells per mil. And this has to be done in a certain length of time too. I think they do it for something like 10 or 15 minutes. And then you make a chart. The next time you want to know the number of cells, you put the cells, and you don't know the number of cells per mil, but you put the cells in something and then measure the CO2 per your unit of time. And let's say it's 10 minutes. And then you measure the amount of CO2, look at your chart to get your number of cells. And that's how you use the metabolic activity. So all future times, you just measure the amount of CO2 from the cells and then look at your chart to determine how many cells you have. And that will be your estimate of the number of cells. Any question about any of that? The dry weight is something similar. The dry weight is often used for filamentous bacteria or molds where it's difficult to do the, I don't know, cell count or the viable plate method. Filamentous bacteria tend to stick together, so the viable plate count will be off. The cell count will be difficult because the cells are all sticking and clumping together. But you could grow up the cells and then measure them, dry them out. So you grow up the cells, pour off the solution after you ultracentrifuge them, dry out the cells, and then weigh them. And then look at a chart for the weight to see how many cells you have. The first time you make the chart, you have to make the chart on, I don't know, something like the weight of 100 cells, the weight of 1,000 cells, the weight of 10,000 cells, the weight of 100,000 cells. So it's difficult to make the chart. But once you have the chart made, you can then simply dry your cells, measure the weight of the cells, look at the chart to determine how many cells you have. Any question about the different methods we have for measuring the number of bacteria? Let me remind you that for all of the methods except for the plate count and the filtration, you are counting both dead cells, like under the microscope, the dry weight, the metabolic activity wouldn't be that way, but the turbidity would be. You're counting the dead cells as well as the live cells. For the plate count, that's the main way we have that only counts for viable cells, although the filtration method and the metabolic activity will happen for only viable cells. Any question about any of that? All right, I'm a little bit past the time, but I finished the lesson. Are there any questions? If not, I'll see you at 6:30 for the lab.